I am excited to introduce Jessica Coakley from the Mid-Atlantic Fishery Management Council, who will be our moderator for the next session. So Jessica, take it away.
Yeah, thank you, Avalon. So today we're going to be talking about environmental DNA and marine systems, applications for the Mid-Atlantic. I'm really excited. We have three excellent speakers for you today. Down below me, we have Dr. Jason Adolph. He's an endowed professor of marine science at Monmouth University, as well as a number of other universities he's affiliated with. He's located in New Jersey. His research interests include phytoplankton ecology and physiology, harmful algal blooms, real-time continuous water quality monitoring, and eDNA approaches to aquatic community analyses. Online, we have Dr. Angelina DiCera. She's an assistant professor at the Batten School and Virginia Institute of Marine Sciences in Virginia, and she's interested in using a combination of genetics, molecular, biochemical, and whole animal techniques to compare how different fish species have adapted to their environments and how they may be impacted by environmental stressors in contrasting ways. Also down below me, we have Dr. Kelsey Leonard. She's a water scientist, legal scholar, policy expert, writer, and enrolled citizen of the Shinnecock Nation. She focuses on indigenous water justice and its climatic, territorial, and governance underpinnings for our shared sustainable future. She's also a member of the Mid-Atlantic Regional Ocean Planning Committee. So for this session, we're going to have a number of short talks that they're going to provide for us. I've got some questions for them as a panel, and then we're going to open it up to the public here today or online for any questions you might have. But first, we have a short video that's going to set the stage in terms of what is environmental DNA. That video was provided by Jan McDowell from the Virginia Institute of Marine Sciences.
Hi, my name is Jan McDowell, and I'm in the Natural Resources section at VIMS, and my background is in fisheries genetics and population genetics, and I've been asked to give you a short presentation today about environmental DNA. So what is environmental DNA? It is DNA that's been released from an organism. So organisms are continually shedding their DNA in the form of things like scales, gametes, larvae, waste products, and things like that. And this shed DNA can be sampled non-invasively from water, soil, or air. And once we have sampled this DNA, we can extract it from the environmental samples, sequence it, and use it to identify the organisms present in the area. And we can do this in a variety of ways that we'll talk about in a minute. But there are several uses for this technique in fisheries biology. So we can use it to, for example, track invasive species, detect rare or endangered species, threatened and vulnerable species. It can also be used to monitor biodiversity, including environmental impacts. So we can track what's happening to an environment that's degrading, or we can track the progress of restoration. We could look at changes in abundance of a species. We can also use it for temporal and spatial monitoring. So for example, in the case of a migratory species, we can track where this species is at a particular time, when does it show up, and where does it show up. And there are a couple of different approaches to doing this. There are actually a lot of approaches, but they can be broken down into two general categories. So we can either detect a single species or a few species. So for example, we could be detecting a species, a genus, or a family, but it's usually a very restricted group. Then we would do this using quantitative PCR, or we could do digital PCR, which is a little more sensitive than quantitative PCR, or PCR-free assays like CRISPR-Cas. And then the second major approach to eDNA is the detection of groups of species, so community-level detection. So in this case, you'll see it called metabarcoding. And metabarcoding can also be targeted to varying groups or to varying levels. So we could target vertebrates with our assays, or if we want to be a little more specific, we could target fishes. And if we want to be more specific than that, we could target elasmobranchs, for example. But the main idea is that we're actually sequencing the entire community of what's in that sample, and then we're searching to databases like either custom databases or National Center for Biotechnology information databases to identify the species that are present in the system based on their DNA sequences. And so there are several advantages to eDNA. That's non-invasive and it's non-destructive. So especially in the case where we're looking at endangered or threatened species, there's no need to handle the species, there's no need, it doesn't get stressed, we don't even need to see it. And we're not harming the environment. So things like electrofishing and dip netting can be labor intensive, they can be harmful to the fish, they can be harmful to the environment that the fish or other species live in. And they're also not as sensitive for early detection of things like invasive species. It's an advantage that we can, all the DNA present in the environment is sampled. So when we take that water sample or that soil sample, all the DNA from all the organisms that have been there in the recent past are sampled. And so we can actually share across labs. So if I want to assay for an endangered species, I can do that assay, and then if I have someone next door who's perhaps interested in doing assays for the invertebrates present in that sample, they can take that same DNA and with their own targeted assay, they can do something completely different. And you can, as I said, you can target different groups of organisms selectively. But there are disadvantages to eDNA, or maybe it would be better to call them caveats. So some of the caveats, probably the biggest one is that we're not entirely clear on persistence in the environment. It's really not understood, well understood, and it can be affected by a lot of biotic and abiotic factors. It can be impacted by species-specific ecology, the metabolic rate of a species, the DNA shedding patterns of a species. It can also be impacted by things like sunlight. How much sunlight is the environment getting? So that will impact the degradation rate of the DNA, the pH of the water, how those water masses are moving, all sorts of considerations like that go into the conclusions we can make with eDNA. It's also less information than traditional fisheries surveys. So I've sometimes heard people say, oh yeah, we could use an eDNA survey and it's just going to replace all the traditional surveys. It's going to be cheaper, it's faster, it's great. Okay, maybe. Maybe sometimes it could replace, in some instances, a traditional survey, but you're not getting the same sort of information. If I take an eDNA sample, I'm not going to necessarily know, do I see evidence of disease? What is that? What are those fish eating? What is the size distribution? What is the reproductive state? Things like that. So it doesn't really replace a traditional fisheries survey, but it's a great supplement. It can require a lot of optimization and many replicates. So when I'm trying to develop, or when someone's trying to develop a targeted assay that, say, only amplifies black-banded sunfish or only amplifies red drum, it may be a lot of work to make sure that it's not also detecting a very closely related species. And depending on how common the species is, so depending on how much of that DNA is in the environment, I may have to do many, many replicates. And that depends on how important it is that I'm right? So how sure do I need to be about the answer? And so for a rare endangered species, if I really need to know, I may have to do many, many water samples to feel fairly sure that it is not, in fact, there. And then the samples have to be filtered and preserved fairly quickly. So you can have things like eDNA backpacks that will filter the water for you, or a lot of the time we're just filtering it ourselves with vacuum pumps. And from the time of sampling to the time of preservation should be about 24 hours or less. Otherwise, the DNA could start to degrade. So just as a final slide, I wanted to show you the general workflow and give you a visual of what the different assay methods look like. And so this is just a picture of a sampler that we sort of made out of a fish tagging pole. So it just shows you it doesn't have to be really fancy. This could be a fancy backpack, or it could be a Niskin bottle that triggers at a particular depth. Or it could be an old fish tagging pole that someone like me taped to the end of a stick. And so we take the water sample, and then we kind of bring it back to the lab, and we filter it down onto these disc-shaped filters, get the DNA. We would then isolate that DNA, and we would either sequence it or we would do a quantitative PCR reaction. So the sequencing is metabarcoding, what we call metabarcoding, and this is an example. So once we have the sequences and we do some filtering and clean them up a little bit, we can match those sequences to a database. And we know, for example, that when we see this sequence, we have a bay anchovy. But when we see this sequence on the bottom, which has differences from the top sequence, it's an Atlantic menhaden. So we're sequencing the community and we're matching those sequences to a database to identify species. In the case of a quantitative PCR where we're doing a single species detection, we want to know, is a particular endangered species detected in a sample? And so if we just say that, you know, each one of these lines is a sample, this is a detection, this is a detection of the species of interest, and these samples were non-detections. So we did not detect any of the species of interest in these samples. And so that's really sort of a visual on the different methods of targeting a community of species versus an individual species. So I hope this was helpful for you, and I hope you have a really great day. Thank you.
Yeah, a lot of thanks to Jan for submitting that to us to share with you. Our first speaker up is going to be Jason.
Good afternoon, everybody. I'm Jason Adolph from Monmouth University, and I'm excited to talk to you about what we're doing with using environmental DNA metabarcoding, mostly in the context of fisheries monitoring plans. Many people went into this talk. I've got 7 minutes. I don't have enough time to sing their praises. This slide's kind of a little outdated. Offshore wind is what got us into this. You can see the plans are very widespread on both the East Coast and the West Coast. And all of these need fisheries monitoring plans to detect potential changes due to wind farm construction. So basically what a fisheries monitoring plan does is ask what fish are in the area where construction is going to take place, and they typically use standard capture kind of techniques, asking questions like what fishes are there and what effects will that construction have on them. There's a list of potential effects that many people anticipate from wind farms, and you can come up with potential effects of other activities, as well as the indirect effects of changing oceanographic conditions. So what we're trying to figure out, and many people are trying to figure out in making fisheries monitoring plans, is what do we have to start sampling now before construction to address this very complex question, because as the wind farms are being built or anything else is being built, there's also natural variability, which is very high in these systems, and global climate change, which is also having an effect on the conditions in the fish populations. So eDNA is here to help, and I put that specifically, as the video pointed out, not to replace things that are being done in fisheries monitoring plans, but to supplement things that are being done in fisheries monitoring plans. This was also pointed out in the National Aquatic DNA Strategy that came out last June, that eDNA is a good use case scenario. We've been through this in the video. This is my slides to show what eDNA is about. I'll just point out in here that the half-life of eDNA in the environment is approximately 1 day. Half of it degrades every day. We filter 1 liter samples, and this is another slide showing the qPCR, single species versus community assays. What I'm talking about today is going to be the community assays or metabarcoding, as was just pointed out. And this is all technical stuff that I'm happy to talk about later. So the work I'm going to talk about is part of the New Jersey RMI program, and the eDNA program that I lead as part of the RMI program has 7 different sampling campaigns that are shown on this map here. There's the offshore wind development area, which is a grid of points over the wind areas here offshore, and the surf zone sampling as well, collected from the shoreline. And the surfline sampling is paralleled by a community effort. We also, community scientist effort. We also pair with 3 ongoing capture surveys, and this is really important in terms of understanding the relationship between what we find in eDNA results and what a trawl, an otter trawl, or a trap survey finds in their assays. And my partner Keith Dunn runs an RMI acoustic telemetry program, another non-extractive technique, and we pair eDNA sampling with that as well. So through this list of projects, we're trying to figure out what is the 2 things. How can we use eDNA best within fisheries monitoring plans to do a better job at detecting change? And what is the relationship between what we find in the genes versus what you would find traditionally using capture techniques? I'm going to go through some results quickly, and we can talk about this more in the discussion area. Each of these plots is a different sampling bout. The green is eDNA, and the orange is trawl. And what we're looking at is how many taxa were found in a sample. And you can see the pattern is that we almost always find more taxa in an eDNA sample compared to its paired trawl sample. Consistently over time. We've done this in other trawl surveys as well. That means we must be catching things in the eDNA that are not being caught in the trawl, and that's what this list shows. The higher a taxon name is on this list here, like bay anchovy, the more frequently we catch it in the eDNA without it being seen in the paired trawl survey. And here's some pictures of those types of fish that we commonly find in the eDNA but not in the trawl. On the other hand, there are taxa that we don't find in the eDNA but that are found in the trawl. The 2 that I want to point out here are 3, pigfish, round scan, black-cheeked tonguefish. They're not found very often. This scale down here on the X is the proportion of times that happens. That's because we don't have them in our reference database. Or I should say we didn't have them in our reference database. We're in the process of updating it, and we've now found, we do have sequences for all of these fish in our database. We just haven't integrated it yet. But that is the drawback of eDNA. If a fish is not in your reference database, you're not going to see it, period. Yeah. So that was trawl comparison. When we compare eDNA with trap surveys on structure, so this is New Jersey artificial reef surveys, the differences are even more stark. We only have one set of data processed here, and shown over here as well. The number of fish that will pot, as they say in trap surveys, that will go into an unbaited trap is very low. But the community composition can be very different around a trap, a structured area, and you miss that in the traps compared to using eDNA. You see how many more taxa we found there. This one here, Cunner, I ran into a diver that works in the New Jersey Artificial Reef Survey, and he said, we see Cunner all the time at the reefs, but they won't go in the pots. You got to tell me if they're in the eDNA. And sure enough, we do detect them in the eDNA. Then this is a pigfish that we also don't have a sequence for. Didn't, but we do now, actually. And then the last piece of data I want to show you is just about one of the powers of metabarcoding. I'm just going to move through this very quickly. Is getting a fingerprint of the community composition through machine learning or other ordination techniques that can be applied and mapped very easily. And you can see here, basically on this map, everywhere the point is a different color, it's a different community composition as determined by eDNA. And if you look closely at these 2 red areas here, those are the artificial reefs, and you can see the different colored points over them where water column eDNA sampling showed a different community composition over those artificial reef areas that you would not have seen through trap surveys. So in summary, we're using eDNA as part of fisheries monitoring plans. We're interested in forming an ecological and methodological baseline. We're very interested in this idea of what are the gear biases inherent to capture surveys versus eDNA and how do they relate to each other. biodiversity is a huge strength, and community science I didn't get to talk about is also a huge strength. Another strength is all these things that you don't see in other capture surveys. The amount of dolphin DNA at the bottom here off the New Jersey coast is huge, on par with some of the more predominant fish, and you never talk about that in capture surveys. So I'll end on that. It's a new set of tools. It's very resilient. It's very scalable. And we're not looking to replace anything in fisheries monitoring plans, but we're looking to supplement those plans with this new tool so that we can do a better job of what we're trying to do. Thank you.
Yeah, thank you, Jason. And I'm glad to see we've got emails and links at the end if folks have questions after this meeting they also want to send to you. Our next speaker is Angelina DeShera. We've got Angelina online.
All hear me? Can you all see the slides?
Yes, we can.
Perfect. All right, I'm going to try to be as quick as possible. As we've already heard, there's a lot of things that you can do with eDNA, and so this is actually the beginning of a project that one of my master's students is working on, Kelly Gonzalez. So this is her work primarily, and we're just at the beginning steps. Because I should say first and foremost that I am a comparative fish physiologist. I'm not an eDNA expert, but my work revolves around understanding how fish do what they do. And so as a comparative fish physiologist, I'm really interested in how different species are surviving and thriving in very variable conditions, very variable environmental habitats. And for the past couple of years, I've been really interested in sturgeon species in particular, starting with white sturgeon in the Pacific Northwest. But as I moved out to Virginia for a position at the Virginia Institute of Marine Science, we have more species of sturgeon out here that we can work with as well. Now, you all might be familiar with sturgeon and know that they have this long history of historical overfishing. So sturgeon are the fish that fancy folks can get caviar from, and so back in the early 1900s when there was a lot of fishing for these fish. When you're taking slow growing, late maturing fish out of the population, obviously you're not doing them a service. And though there's lots of laws and regulations in place now to protect sturgeon from fishing, they still encounter lots of things like habitat alteration and degradation, so such as dams and their migratory corridors that could prevent just a physical barrier that could prevent their movement, but also changes the quality and chemistry of their habitat downstream. And as a physiologist, I'm really interested in things like global climate change. And so heat waves are becoming more frequent, more intense, longer duration, as we're thinking about ectotherms that can't control their internal temperatures. This is really important. And you can see in the Pacific Northwest here, what looks like little veins, those rivers are getting really hot really quickly within a short period of time. And so understanding how these fish are functioning in their habitat is really important. And we've noticed from the literature, one of the big questions that we still don't have a good idea of is how sturgeon, if they have suitable habitat that matches their physiological demands and limits. And there is this gap between field data and in-lab data where critical temperatures for presence, so where these fish are found and where they're spawning, is actually typically lower than what we are looking at in the lab. And so understanding what that mismatch might be and why they might choose different habitats. So when I moved out to VIMS, I was interested in our local population of Atlantic sturgeon in particular. We have the Chesapeake Bay distinct population segment where the James River is a well-known spawning habitat, a critical spawning habitat, but there's evidence of spawning in other rivers and tributaries in Chesapeake Bay as well. So here, the star right here is Gloucester Point, where I'm located at VIMS. We do see in the Pamunkey River, there's evidence for spawning as well. And the particular tributary that we're interested in right now in my lab is the Chickahominy River. So sturgeon have been reported at the mouth of this river, which is a tributary of the James River, which is a known spawning habitat. But we have really no idea about their use of this habitat. So we want to understand what factors might drive their presence or their habitat preference. So very simply, we're looking to investigate first and foremost, where are they and how do we find them? So that we can then follow up with some more specific physiology questions. Traditionally, sturgeon have been found through fishing efforts, tagging efforts, sonar. You see these cute little images right here of a sturgeon that you can see visualized from a boat. But as Jen mentioned in her video, these techniques are especially difficult when you're working with threatened and endangered species, where there are very few of them, so your effort is really high. So this is where my interdisciplinary team comes into play, including Jan and Ellen, her senior research specialists that are eDNA experts, so that we can use a different technique that is a little bit more cost and time effective for us to look for these sturgeon. We're also collaborating with our habitat experts in the Chickahominy Indian tribe, Joe Hunt, Dana Adkins, and Wesley Adkins, that understand this habitat and are helping us to identify different characteristics of the habitat quality and chemistry so that we understand a little bit more about what may drive preference or avoidance of this habitat. And then Kelly Gonzalez is my master's student whose project this is, and so she's a soon-to-be eDNA expert. As was already mentioned, we are using a very specific type of eDNA analysis. So where we're using qPCR, where we're just quantifying specifically Atlantic sturgeon DNA to understand their presence and absence, and hopefully abundance as well over time. As you might imagine, for finding threatened and endangered species, this is an excellent tool for finding that needle in a haystack for Atlantic sturgeon in these tributaries. And the good news is that eDNA has already been used successfully to search for sturgeon, both locally, so in the Pamunkey River and the Nanticoke River. They have found presence of eDNA presence in these rivers, but it's also been such a useful tool that it's been used in outreach and education initiatives in the Hudson River, where high school students were able to collect samples, filter samples, and then send them off to collaborators to look for Atlantic sturgeon eDNA. So right now we're just at the beginning steps where we're searching for sturgeon, and we are identifying the sites that we're going to be looking for these sturgeon along the Chickahominy River, where site 1, 2, and 3 are along the property, the ancestral land of the Chickahominy Indian tribe, the Manahunt, and the mouth of the Chickahominy River, where Atlantic sturgeon have at least been visually sighted before. Kelly is taking samples both in the shallow and deep habitats. So because these fish are benthic, trying to understand first our methods of where are we going to find the highest abundance of eDNA. And then we have a couple other sites along the river that are we think of as our positive controls. Because the James River is a known spawning habitat of Atlantic sturgeon, we would expect to see Atlantic sturgeon DNA at least sometimes during the year in these sites. And so right now, Kelly is practicing with a couple of preliminary samples that we have taken in October and April of this year. And the hope is that we can establish some of our methods, figure out how much samples we need in order for us to start in the summer, to start a periodic monthly sampling to see if we can find seasonal and spatial presence of Atlantic sturgeon. And then, of course, my personal hope for this project is it's going to lay a baseline for our understanding of habitat use of sturgeon, whether they use the Chickahominy or not. Understanding the habitat quality there as well might give us some insight to why they might prefer different habitats for spawning. And then understanding down the line if there's a mismatch between spawning and successful recruitment. So is a habitat suitable for spawning, or is it suitable for rearing of young sturgeon as well? And that's it. Thank you.
Thank you, Angelina. So thank you, and just hang on. Online, we'll have some questions coming up. Our next speaker is Kelsey Leonard. And I'll note, Angelina, I did see Jan's pole taped to the Nalgene bottle in a couple places in your talk, so she's putting that to work.
I'm going to present from down here. Saves us time for Q&A. So thanks, everyone. I am just so pleased to be a part of this panel, and I hope I can kind of round us out with some of the components that my colleagues, I think, were alluding to about new technology, piloting, and testing. And so that's what I want to share in the context of eDNA and our Shinnecock Hope Spot expedition that we did this year. So in June 2024, this project that I'm sharing with you today is a collaboration between the Shinnecock Nation, the New York Explorers Club, as well as Mission Blue and their Hope Spot program. And in June of 2024, with sirens, in June of 2024, we received an Explorers Club expedition flag to run a year-long expedition focused on environmental DNA in the Shinnecock Bay. And so you can see Sunshine Gums from the Shinnecock Nation and Dr. Ellen Pickett receiving the expedition flag at New York. If folks are unfamiliar with the Explorers Club, it's a very prestigious institution that has supported expeditions around the world, and some of our most, I guess, magnanimous scientific explorations in the past 100 to 200 years have been done under their flag. So really excited to be able to bring an Indigenous expedition to their work. As I mentioned, this was in partnership with Mission Blue because in June of 2022, Shinnecock Bay, which we are named for, or the bay is named for us, which our territory as a nation is along the bay, it was named Hope Spot by Mission Blue. And these are special places that are identified as being scientifically valuable to our understanding of ocean health. And they're championed by local conservationists, and in this particular instance, by Dr. Pickett, myself, and the broader Shinnecock Nation. Oh, a little bit of, nope, it's cut off. Well. Anyways, this is from our first gathering of our Indigenous Advisory Council, which we also set up in tandem with the establishment of the Hope Spot to allow for this committee of Indigenous advisors to inform our scientific research, exploration, and design. And so this was that first meeting, and sort of our research team facilitators featured here. So what is the IAC, as we like to call it? It's a committee of Shinnecock tribal citizens who are interested in an advancement of scientific research in Shinnecock Bay. As we mentioned, it was formed after the Hope Spot in 2022, and its overall goals are around environmental stewardship, protection, and scientific leadership. To date, we, in conversations with Mission Blue, the Shinnecock Indigenous Advisory Council is the first Indigenous Advisory Council for a Hope Spot. So we're really sort of at the cutting edge of seeing how we can allow for Indigenous knowledge, Indigenous science to inform ocean conservation and ocean science more broadly. And this is some of the visual notes that came out of our conversation with that advisory council in our first meetings in 2024. This was really our visioning document for how we anticipated the collaboration around the expedition to occur. And in large part, you can see there eDNA sort of at the center in that water droplet. So we successfully were able to deploy our expedition. It began in September of 2024. And what you see here is our larger research team, as well as with Dr. Sylvia Earle, who, if folks are unfamiliar, is the founder of Mission Blue, and the Mission Blue Hope Spots is an idea that she has championed and sort of originated. We also have a colleague from Ocean Robotics who helped us to put forward the USV, which is an uncrewed vehicle that you see there, surface vehicle, and this was paired with what we call the Roxy, which is an eDNA sampler. So it's also completely solar powered. So we were able, and this was the first time that this technology was ever being deployed in U.S. waters, and particularly as matched with an eDNA sampler. So what was really exciting for us was to be able to deploy this USV. It went around Shinnecock Bay collecting over 100 different, from 100 different sample sites to then allow for us to add to our biodiversity assessment. So wonderful video that Jan did earlier. So we do meta-barcoding. So we're really looking at establishing that community assay for our baseline biodiversity assessment of Shinnecock Bay. Why this is also really important here is this is solar powered. So we're also trying to, again, think to Jan's earlier video of what can be the least impact, least destructive way in which we can understand biodiversity in our habitat. So this is the Data Explorer is what it's called, paired with the Roxy with the expedition flag on its back as it is going through Shinnecock Bay. So it's an integrated solar-powered USV with an autonomous eDNA sampler, Data Explorer and the Roxy. It really was allowing for Shinnecock Bay as a hope spot to do what it was intended, to be that sort of test bed for scientific exploration and to see what the potential of this new technology is for our communities. So just kind of rounding out, this is some of the Shinnecock Nation participation. So we had many different tribal citizens participating, loading the USV off the dock and into the water, manning it. It's just kind of like a plane almost. You have to have a pilot who is monitoring it 24 hours as it's cycled throughout the bay. Now, what I want to end with everyone on is just also in this research design process, how we think about Indigenous data sovereignty in the collection of environmental data, and particularly eDNA. So myself and others, you may know about sort of the FAIR principles for data. We put forward what are called the CARE principles for Indigenous data governance, which are now pretty standard across most countries, particularly Canada, New Zealand, Australia, and the United States. You may see them be referenced in NSF applications, depending on where that goes in the world. But what we also are very proud of with this project is we utilize local context, which is an entity, an organization that allows for us to do within the metadata of environmental data that we collect, we're able to use notices to notify Indigenous nations, Indigenous peoples whose data we are collecting, because all data within Indigenous territories is Indigenous data. And so this is sort of the impetus or the impetus and the expression of why local context is important. So these are what some of the notices look like. There are biocultural notices, traditional knowledge-related notices. And so I also just wanted to shout out a few programs that are doing really cool eDNA work. And to my earlier colleague about working with the Chickahominy, I actually see a lot of Indigenous nations leading in eDNA. And I think it's to what Professor McDowell said earlier in the video. We are really tired of trawling. And for my colleague, who we partnered with, Dr. Ellen Pickich at Stony Brook University, they were sort of forced to start collecting and doing eDNA work in 2019 because we had hard clam bed sanctuaries put in place throughout Shinnecock Bay. So you couldn't trawl in the hard clam sanctuaries. So how else were you going to do those surveys? And so they went to the supplemental of eDNA, and then we saw that as a nation, and we said, hey, you should be doing that everywhere to not impact the habitat as least as possible. And so Maine is doing this at the University of Maine, and they partnered with the Wabanaki Confederacy. They have a memorandum of agreement through their regents at the university to collaborate. And so this is just a bit about Maine eDNA, and you can find some of their papers out there. Obviously, the wonderful work of Chickahominy. We had some students in my lab map out some of the different types of Indigenous-led eDNA projects, and this data was from 2022, so now we've got the Chickahominy project and so many more that we can add to this, but you can see that globally, there's not that many eDNA projects. So to see that you've got, close to 10 different ones that are Indigenous-led eDNA projects, that's pretty cool. So just to close out, I just wanted to again kind of reiterate that in the context of environmental data and eDNA, this is Indigenous data, and that's probably why you are seeing these knowledge experts like with the Chickahominy, like with us at Shinnecock, really being tagged in to create a very blended and braided science approach. So our results of our biodiversity assessment, the samples collected in the fall of 2024 will be released in this coming fall of 2025. And this is just more of the Indigenous Advisory Council with the USV at its deployment in September. So thank you so much, and turn it back to the group.
Yeah, thank you, Kelsey. So if we could just pull Angelina back up on the screen. I just have 2 quick questions for each of our panel members, really about opportunities and challenges with eDNA approaches, where are the opportunities for growth, to support regional marine and coastal decision-making in terms of the UCE and eDNA, and some of the challenges you see to integrating these types of approaches in the region. So if you want to just touch on that quickly, Kelsey.
Yeah, I think the easiest answer is I think there's so many opportunities for tribal participation, tribal leadership, indigenous leadership in eDNA. So if you have an eDNA project or are considering one, thinking about ways that you can scale and pool resources, not only with tribal nations, but with others. I think citizen science was mentioned earlier, educational programs. I really, there's not a better way to say it other than the sky's the limit with eDNA, just because of what can be minimal cost. I will say the Data Explorer and the Roxy we benefited from a lot of donations, but I'm hoping as more people invest in eDNA, we can scale the cost of the new technology down to make these systems very accessible for a wider array of the community.
Yeah, thank you, Kelsey. Angelina, do you want to weigh in?
So I think what Kelsey said was perfect. That is how we have been thinking about eDNA, is how can we broaden the participants that are collecting this data. And it is a really easy way. It's an easier way than asking folks to trawl with us to participate in science and actually see results in real time, which is really exciting and a way to get buy-in from a local community. So it's very easily accessible, which we love.
Yeah, thank you, Jason.
So I'll echo, I really like the community science applications of eDNA and getting people involved in fisheries sampling. Everyone's got a bottle, not everyone has a trawl ship. And it opens a channel for outreach and communication between the scientists and the community as well. But I'm also going to add the growth opportunity is sampling from AUVs, the way Kelsey described. For the offshore work that we do, it's a little more challenging. We would need like a 2,000-kilometer track in like 2 weeks at sea kind of thing. And we're talking to people from Mbari and working with Rutgers to adapt some of their gliders to be able to do that. But it all comes down to, I think, the scalability of eDNA. That's one of its greatest strengths. The more we can exploit the scalability of eDNA to get more data from more areas, I think the better we'll be doing at using it best.
Oh, great points. So those are my questions, opportunities and challenges. So why don't we open it up to the room or to online participants to see if they have any questions for any of our panelists.
Oh, I see one. Ryan?
Hi, Ryan Green, Virginia Coastal Zone Management. I'm wondering, this is something that we started thinking about when we first learned about eDNA, and then we realized probably it's fraught with more challenges, but are you guys aware of where eDNA has been supplemented or used to support compensation, fisheries compensation, or other kind of hard environmental management, frameworks, things like permitting, anything like that? I know we're early days, but it seems like eventually we could get there, and I wonder if you guys are aware of anywhere that's kind of starting up.
I can't think of anything per se, but I do think they're not as, I think, firm as you're articulating it. I think within Shinnecock Bay, there's been, as I mentioned, the synergy with the hard clam protection beds, then what we call sort of hard clam sanctuaries, that now that, trawling is still done, but to allow for us to have a proof of concept of the eDNA results so that there's a hopeful phase out of trawling. And that is making a better case for us to maintain these sanctuaries, because we are seeing population restoration and rebounding where those sanctuaries have been. And so I think less firm than what you're articulating, but I do think it is helping to support the idea that having sanctuaries in place is really helpful for population maintenance, and then there could be rotating sanctuary areas that allow for those to be opened up for fishing, and then another area that's in need of restoration gets placed. And then to protect those, you have this eDNA process that allows you to still understand the biodiversity of the area. My colleagues may speak to this as well. Like, a challenge, I will just be honest, is abundance. We don't really have abundance data right now. What we have is absence presence, which is still very good for a baseline biodiversity assessment, but we do need the technology and the science to continue to be invested in so we can get to a metric of abundance.
Do you want to add?
I would look into what David Lodge did with, I think it was Asian carp in the Mississippi River, which is kind of like the OG fisheries eDNA work. But I'll add that the National Aquatic eDNA strategy that was put out in June last year, one of the reasons that was put out was that people are reporting issues in permitting with federal agencies, in that some will accept eDNA as data. There's no legal barrier to accepting it, but some will and some won't. There was no guidance from the top indicating that, yes, this should be used as data for those purposes. So I think that's where the state is now. I think it's kind of on the fence. And I also think that the offshore wind use of eDNA, along with other fisheries monitoring plans, will go a long way towards further establishing eDNA as an acceptable form of data for permitting and decision making.
I think that's actually a really important point that I should note too, that also brought our marriage with sort of between the Shinnecock Nation and Stony Brook University, is Stony Brook University could not sample in our territorial waters. And then if you are working with other tribes, like we have our own permitting processes and things. And so I'll just speak from our jurisdiction, our regulations, we don't tend to take kindly to permits of trawling, especially if it's going to impact our local fishers and bay folks, because they're like, no way are you taking a trawl near my oyster beds. So with that, we are able to find a better marriage with the science to say, we will allow for permitting of sites and samples if you use a less invasive methodology like eDNA. And so that's where I'm seeing tribal nations be more open to permitting and regulation in this space.
Other questions?
So since one of the speakers introduced a different geography, I feel like I can talk a little bit about the Great Lakes. I'll say, the eDNA conversation first came to my attention with bighead carp and some of the concerns about release of Asian carp into the Great Lakes system. eDNA was used fairly early on, at least in my experience, in trying to detect carp below physically or hydrologically separated areas that were still connected somewhat as waterways. And I think the experience that we had in government at that time was, is the eDNA really showing us that the organisms are present and they were detected in areas where previously nobody thought they had invaded yet? Or are we getting some sort of like background sampling? And I'm just wondering if you can speak to any advances in the technology where you can determine, like, are we really seeing something that's there or something that's upstream or something that was elsewhere in a hydrologically connected area and somehow that DNA kind of found its way into this water body? Just as a, again, are there advances enough? I heard speaking to the half-life of, 1 day. So are we talking about, the size of the sample of eDNA that you're using can tell you about how old the sample is, or are there any other kind of cues you can use to determine whether it's truly a presence thing or if it's just kind of backscatter, background, whatever you want to call it?
Well, given the sturgeon hunt that Angelina is on, do you want to take that one?
I was going to say that is probably one of our biggest challenges right now as we're starting up our sampling is because we don't know how long sturgeon eDNA stays in an environment, and if these fish are really highly migratory and very transient in a place, we might have the opposite problem where instead of us picking up a signature of a sturgeon that is in a different area, we might miss the signature of a sturgeon because the eDNA is degrading so fast that if we're sampling once a month, we're not going to capture that signature. So I'm actually typically thinking about it in the opposite way, where I am more worried about eDNA not lasting in an environment versus it being like traveling from a different environment and being picked up elsewhere. That is our current challenge right now.
Yeah, thanks, Angelina. Do you guys have anything you want to add, or?
It's the question that always comes up. And there's a lot of work that people have done, like putting fish in a tank and then removing them and then counting the time as the DNA goes away, kind of experiments. That's where the half-life I cited comes from. And I agree with Angelina. I'm more concerned about missing eDNA because I sampled too late than sampling eDNA that represents a fish that was present 2 weeks ago. Because the former is more likely, the latter is very unlikely. And that said, we do see, but the transport issue is very important too. One of the first eDNA results I presented at a fisheries meeting showed mummy chug offshore in the Orsted Ocean Wind 1 wind area, 20 miles offshore. Mummy chug is a coastal estuarine fish. And a hand went up immediately. Or maybe Motts came up to me afterwards and said, why are there mummy chug there? And you know, fish genes are different from fish. A fish gene can make its way 20 nautical miles offshore. You can think of a lot of ways that could happen. But a mummy chug probably wasn't there. So it takes a different mindset to interpret eDNA data than trawl data. It was a trace mummy chug signal, but it was still present. So they're real issues. And those are the kind of things that we're working through. And in my opinion, why doing a lot of pairing with capture surveys is really important to really understand what the eDNA signal is telling us. And we're lucky to have all the New Jersey capture surveys that are ongoing anyway. We just tell them to collect a bottle and send it to us.
Yeah, I think a lot of it is really cost driven as well. So, I mean, that was one of the, I think the key takeaways from our work in Shinnecock Bay is when you have this USV paired with the Roxy, it is doing continuous sampling over a 24-hour period. So to what Jan said in the video of the optimization and the replication, the hope is that with that, we can have more confidence in the results that come back. So that's a little bit of our approach. But yeah, it's less so of are we getting something from very far away, but rather than are we missing something in the 24-hour period that we know it could degrade in.
Okay, thank you. It looks like we have time for like one more, Avalon.
Yeah, I was going to ask, do you have a correction for a dead fish that could be swept away into different water bodies?
Still DNA.
It's all the same DNA. I didn't see who asked that question, so I can't look at you. Dead fish is going to have the same DNA signature as a live fish. So no.
Yeah. Okay. And I think that gets to additional investments in the science so that you can perhaps have something that allows for control with that later on, but then it's also an abundance question, right? So abundance is also about, you know, how much is there and is it alive when it's there?
Yeah.
Great. Well, the clock tells me I'm out of time for this session. So I want to thank all of our contributors, starting with Jan, Jason, Kelsey, Angelina for hanging in there online and joining us. Many thanks. And I think you'll be around in the room later today if you have any questions. And I think in the talks, Angelina also has her email and contact information. So if folks can reach out if they have additional questions as well. So, all right. Well, thank you to our presenters.
Thank you.